My name is Partha Majumder. I am a professor at the Indian Statistical Institute and a founder of the National Institute of Biomedical Genomics. The Indian Statistical Institute is in Kolkata and the National Institute of Biomedical Genomics is in Kalyani, which is close to Kolkata. So, as we know that human populations are structured in many ways. First of all, we are socially structured. We tend to choose mates from our own groups, and these groups are based on certain cultural, linguistic and other parameters. Because of these kinds of groupings, the genetic variation that arises within a group over an evolutionary period of time tends to remain within that group. However, we are not a completely closed group. We also choose mates from other groups, and so there is gene flow from one group to another. The gene flow is usually much smaller than the extent of genetic variation that is resident within a community or within a group. So, over a long period of time, there is some amount of admixture of genes from one population to into another population. However, like I said, the genetic variations that arise within a population group tends to remain localized within that group. As a result of this, as a result of human populations grouping themselves and genetic variations that newly arise tending to remain within the group, between groups there is a lot of genetic variation that one obviously sees. There are two or three things that we need to do. We need to understand where this variation comes from. We as a species, we arose in Africa and moved out of Africa to populate other parts of the world, and as we moved out, there were smallish kind of groups that were formed. There was little amount of admixture to begin with. Over a period of time, this has expanded, and these genetic variations need to be studied. These genetic variations also lead to different kinds of susceptibilities to diseases, responsiveness to various kinds of drugs, etc. This is not to say that genes are the only factors that are responsible for providing susceptibilities to diseases or to the maintenance of health or to responses to drugs. Environment also counts, but the genes and environmental factors, they interplay in different kinds of ways, and we need to understand those. To be able to understand those, we need to use statistical methods, because after all, we are looking at group averages or group constellations or group characteristics, and to be able to find group characteristics that are different from one group to another, we need to use statistical methods. Over a period of time, what has happened is that the cost of identifying genetic variation has fallen, and so we are able to find more and more genetic variation that we are able to capture from within a particular group and also between groups. As a result of this availability of larger and larger amounts of genetic variation, we are better able to find genetic relationships among groups. We are better able to track migrations from one geographical region to another, and as a result of that, we are able to explain far better what these genetic variations, or how these genetic variations have arisen, and what the genetic structures of these populations are. In terms of identifying the genetic variation and correlating them to health and disease parameters, what we've been doing is to study specific diseases that are of interest or that are highly prevalent in different geographical regions, and take a bunch of individuals who are patients afflicted with the disease, and take another bunch of individuals matched for example, for age, and who are not afflicted with the disease, and find if there is a genetic contrast between the two. This is what we call as a genome-wide association study, or a genetic association study more simply. Using this kind of an approach of studying a group of patients, looking at their genetic characteristics, and looking at a bunch of normals and looking at their genetic characteristics, contrasting these will provide us with information, or have provided us with information about which particular genetic variants are associated with a particular disease. Once we find that association, then we get into the lab and figure out what these genetic variations do at the cellular level, at the molecular level, and how we can relate that to various diseases. And that's how we identify biological pathways, starting with genetic variations, identify biological pathways, and then figure out whether those biological pathways can be manipulated by small molecules or peptides or whatever in order to find new drugs. So genetic variation not only enables us to answer questions such as where do we come from, how have we migrated. It also enables us to understand how various kinds of diseases and health parameters can be maintained. Various kinds of diseases, we become susceptible to various kinds of diseases, and eventually it can lead to providing information about drug solutions or treatment solutions for various diseases. In that context, what we've also been doing is we are looking at specific diseases. In particular, our lab has been looking at head and neck cancers, primarily oral cancer of a particular kind called gingivobuccal oral cancer. We are doing this in the context of an international consortium called the International Cancer Genome Consortium, or ICGC in short. And the reason why we are looking at the gingivobuccal oral cancer is because in the oral cavity, gingivobuccal oral cancer, which is essentially all of the oral cavity except the tongue, is very, very frequently affected in Indian oral cancers or oral cancer patients in India. When we contrast this with the oral cancer that's found in the Western populations, it's predominantly the tongue in the oral cavity that is affected in oral cancer. One reason why gingivobuccal oral cancer or the cancer of the oral cavity is so common, not the tongue but the other parts of the oral cavity, especially the lips, etc., are so common is because of the way that tobacco is consumed, chewing tobacco, where one uses lime, which essentially chews up the epithelial lining, and then the tobacco carcinogens directly hit the cells and produce tumors. What we've been looking at is cancer, even though we think that it doesn't run in families, only about 10% of cancers run in families. Cancer is a completely genetic disease. If you look at it at the cellular molecular level, there are genetic changes in the tumor tissue, and therefore even if you are not born with a particular genetic change, you acquire those genetic changes during your life course. These are called somatic mutations as opposed to inherited mutations that are called germline mutations. So what we've been looking at are these genetic changes that arise during the life course of the individual, resulting from various kinds of exposures, such as in particular tobacco exposure for oral cancer, chewing tobacco exposure for oral cancer. What we really like to do is to figure out what are the genes that get altered recurrently or frequently, genes that drive a cell to becoming a cancerous tissue in the oral cavity. We've been doing that. More recently, we've had some good publications, good interesting findings. We've been able to identify 10 genes that drive cells in the oral cavity to becoming a tumor upon acquisition of changes. And these are called driver genes. So we've been able to identify driver genes in oral cancer. These driver genes also have certain kinds of characteristics, such as in cancer there are two classes of genes that get altered. One's called the oncogene. One class of genes is called the oncogenes, and the other is called the tumor suppressor genes. In oral cancer, what we've been able to find is that it's predominantly the class of genes that are called tumor suppressors that get altered, and they drive cells to becoming malignant and producing the tumor. We've also found some evidence of a specific biological pathway being altered, which is called the arachidonic acid metabolism pathway. The arachidonic acid metabolism pathway is very interesting because there are inhibitors that are already available in the market, and therefore it's quite possible to repurpose those drugs for treatment of oral cancer, and such is the thinking in the community among clinicians right now. In doing all of this, we have to use statistical methods, we have to innovate new statistical methods, and I've been involved in devising many of these statistical methods that are used both from the point of view of understanding how genomic variation arises, how do you quantify genomic variation, how do you measure similarities among populations on the basis of genomic variation, as also trying to find genes that drive cells to becoming a malignant tumor. So this is what I've been doing, and this is what my contributions have been predominantly.